Today, we are talking about a medical breakthrough that is happening right now, one that's about to change millions of lives. India has just developed an incredible, revolutionary cure for a devastating genetic disease, and we're going to break down exactly how they did it. So here's the big news. India has officially launched its very first homegrown gene editing therapy. You guys, this is a huge deal. It's offering a potential cure for sickle cell disease, which is an inherited blood disorder that affects millions of people, and it's especially common in India's tribal communities. This isn't just some science headline. For so many people, this is a lifeline. All right, let's dive right into this incredible development. The therapy at the heart of this story has a really powerful name, Bursa 101. This whole thing is the result of a powerhouse collaboration. It was developed by the brilliant minds at CSIR-IGIB, and it's named Birsa 101 in honor of the revered tribal freedom fighter Bhagwan Birsa Munda. And that's a really fitting tribute, given how much the disease impacts these communities. Plus, they've partnered with the Serum Institute of India, so the goal is crystal clear. Make this cutting-edge treatment not just work, but make it scalable and affordable for the people who need it the most. So this therapy is a massive leap forward. But it all comes down to this really ingenious piece of biological tech that has completely changed what's possible in medicine. So how does it actually work? What's the secret sauce? Well, that technology is called CRISPR. You've probably heard the name buzzing around, but what it does is honestly nothing short of revolutionary. It gives scientists the power to literally rewrite the code of life itself, DNA. Okay. Yeah, that's a mouthful, isn't it? Clustered, regularly interspaced, short palindromic repeats. But don't worry about the long name. The key thing to remember is that CRISPR is a tool that lets us edit DNA with incredible precision. The best way to think about it is like a find and replace function, but for our very own genes. And here's the wildest part. We didn't even invent this from scratch. We just found it. See, CRISPR is actually an ancient defense system that's found in bacteria. They're nature's own original genetic engineers. It basically works like this. When a virus attacks a bacterium, if that bacterium manages to survive, it actually captures a little piece of the virus's DNA. It then stores that snippet, almost like a genetic mugshot. So the next time that same virus shows up, the bacterium uses this memory to instantly recognize and just slice up the invader's DNA, neutralizing the threat. Pretty smart, right? So how did scientists manage to turn this ancient bacterial immune system into a tool for us? Well, it's pretty clever. They create a custom guide that matches a specific gene they want to edit. Then they attach that guide to a special protein and pop it into a cell. From there, the system just does its magic, finding and cutting the DNA at the exact spot they targeted. The first key part of this toolkit is a protein called Cas9. The easiest way to think of Cas9 is as a pair of molecular scissors. I mean, they're incredibly precise. It's the tool that does the actual snipping of the DNA strand, but it doesn't just cut randomly. And that's because it has a partner, the guide RNA. This little piece of RNA is basically the GPS for the entire system. Scientists can program it to match any DNA sequence they want. It then hooks onto the Cas9 protein and steers the scissors to the exact spot in the genome that needs a little snip. It's a really, really elegant system. Now, CRISPR-Cas9 gets all the fame, and for good reason, but it's important to know it's not the only game in town. It's not the only tool in the gene editing toolbox. Scientists have actually developed several different ways to do this kind of delicate work over the years. As you can see, there's a whole bunch of them. You've got older tools like zinc finger nucleases, or ZFNs, and TALENs, which use proteins to find their target. Then there are meganucleases, which are natural enzymes that can chop up really long sections of DNA. But what makes CRISPR such a game changer is its use of that RNA guide, which just makes it so much easier to program and incredibly versatile. So what's next? I mean, the story definitely doesn't end with Birsa 101. In fact, scientists in India are already way ahead of the curve, developing the next generation of gene editing tools and really pushing the boundaries of what's possible. So meet TnpB. This is a brand new genome editing tool that's being developed. And what's so cool about it is that it's based on something called transposons, which you might know as jumping genes. They're these little bits of DNA that can literally move themselves from one location to another inside the genome. And here's why that's a huge deal. TnpB is much, much smaller than Cas9. It's tiny. And because it's so compact, it's way more agile,
and a whole lot easier to deliver into cells, especially plant cells. This could totally open up huge new possibilities for agriculture. But the innovation doesn't stop there. Here's a thought for you, and this is pretty wild. What if you didn't have to just trust that the gene editing was working? What if you could actually watch it happen in real time?
Well, guess what? It's not science fiction anymore. Scientists at the Bose Institute in Calcutta have developed something they call GLO-Cas9, and it does exactly what it sounds like. It's a version of the CRISPR protein that literally lights up while it's busy editing genes. So how on earth does this work? It's magic, basically. The team fused the Cas9 protein with fragments of something called nanoluciferase, which is a light-producing enzyme they borrowed from a species of deep-sea shrimp, of all things. When the GLO-Cas9 protein is actively working and editing a gene, these little fragments come together, reassemble, and start to emit a visible glow. It's a real-time signal that the edit was a success. How cool is that? So the bottom line is this: from developing affordable cures for diseases like sickle cell to engineering proteins that actually light up while they work, this technology isn't just theoretical anymore. It's here, and it is fundamentally reshaping our world and everything we thought was possible.
And that leaves us with a pretty profound thought. We now have the tools to edit the blueprint of life itself. That power is in our hands. So the only real question left is, what story will we as a species choose to write next?